Okay. So good evening and welcome to today's Asset Colloquium. It's titled Affordable Healthcare Technologies in Sensing, Drug Delivery and Therapy. I mean, given the current situation for the couple of years, I mean, I don't have to overstress the importance of such a talk, but also I think it's even more so that we are going to have this talk from Professor Rohit Srivastava from Department of Biosciences and Bioengineering from IIT Bombay, who is the Sant Swarup Bhatnagar awardee this year. And to just say a couple of words about him, is of course well recognized for his translational research in the field of biosensors and affordable point of care diagnostics technologies, particularly for rural and maternal health care. I mean, one large number of awards and fellowships, to just name a few. As I mentioned, the Sant Swarup, the INAE Fellowship, Abdul Kalam Technology Innovation National Fellowship, NASI Fellowship, both FRSB and FRSC, the Om Prakash Bashin Award for Excellence in Health and Medical Sciences, the DBT Tata Innovation Fellowship Award, the DBT Process and Product Commercialization Award, NASI Reliance Industries, Platinum Jubilee Award, the Wasvik Award, the DBT IYPA Award, and DBT National Bioscience Award. I mean, quite a few of them to kind of name Professor Srivastava's accomplishments for the over last 15 years. In his career at IIT Bombay, he has graduated more than 35 PhD students, about 80 plus MTech students, and supervised more than 100 research interns. He has published over 200 reputed journal publications with an H-index of 37. And he also been very active in patents, and he was granted 24 Indian and US patents, and additionally filed about 100 plus patents, trademarks, and IDFs, and so on and so forth. And he has also mentored a large number of startups, particularly in the area of medical technologies, for the over last 10 years, and helped them to secure grants and develop innovation solutions for healthcare. So needless to say, we have a really very accomplished and most distinguished speaker to talk on this topic today. So without taking more of the speaker time, let me invite Professor Rohit Srivastava for his asset column.
Yeah, thank you very much. Thank you so much for the invitation. And I consider this an honor to be able to speak at this colloquium. Let me first share my slide and then I'll start. So I hope you are able to see my slides. I'll put it on full screen mode. So once again, thank you very much and welcome to everybody. As already, given in the intro, I joined IIT Bombay in 2005 and became a full professor in 2015, and then took up the charge of the department in 2019. And that is when I had to move a lot from my lab into administrative work. Although I did not stop doing translational work, as you will see shortly. So my lab is called Nanobios Lab because we started off using nanotechnology and biosensors, but then we forayed into many areas, which I'm going to talk about today. Now, if I just give you, so if you look at this slide here, I start off with this slide. Why? Because I would like to emphasize that we sitting in biosciences, bioengineering are not complete. Unless I collaborate with all these other engineering departments, not all of them at the same time, but one at a time, two at a time, whatever. And some of these science departments, I will not be able to do a project or take a project all the way from ideation stage to the bedside. And my goal is always, with any project that I pick up, my goal has always been take it from that idea, which is TRL1, all the way to TRL9. And I'm going to show you several examples of how we've been able to do that. Many of these are TRL9, many of these are TRL4, 5, many of these are moving ahead from TRL1 and 2. So, when I say TRL, technology readiness level, which is a really nice way of assessing how technology moves from all the way, from just an idea in your head to somebody's bedside where it is being used. So that's how we see technology. And when I want to do this, I would like to use expertise in different departments. I cannot do all of it myself. So if I, being an engineer, tie up with somebody, from the medical institute, or tie up somebody from mechanical engineering department to do the, to give me an idea, uh, think of a solution to that idea, and then think of design for that idea. This is where I need all these engineering departments. And then of course the science departments comes into play when I would like to have some more inputs into the solution that I'm thinking for that idea. And this is where, again, once you have a team ready, once you have the idea and you think you have a solution for it, you need public funding or private funding. And we've been very successful getting those. I'll again talk a little bit more about that. But once you have this in place, you now start thinking of how you would like to take this forward into commercialization. And in most of these cases, we usually tie up with a student company right from the beginning. I do not bring a student company somewhere in the middle, or I do not talk to a company somewhere in the middle, because I feel that if you involve a company right from the beginning, they will give you valuable inputs to take a product all the way into the market. And that's where I say, these red and green blocks at the base, either you have your own startup company or a student has a startup company, or you actually do a technology transfer to the medtech industry. Once you do that, then you are definitely going to see the product in market in some time. And these are all indigenous products made in India. These are going to create an impact in this market because, most importantly, our criteria is affordability. I do not care many times about whether that's the only product being made in the world, because many times India lacks in very, very simple technologies, and I'll come to those very shortly. But if I just give one example of affordability, and affordability is something that really brings a product to the market, and this country needs affordability. I'll show you again some examples of those. In that, you are now going to create high value jobs, and you are going to do some capacity building. And we do all of it because, I don't believe in going halfway. I always believe that if you have a student company who's put that product in market, now how can you help the student company? How can you help that company raise money from investors, raise money from forums like BIRAC, which actually fund all these companies, or raise, dilute themselves and raise money at different levels so that they can keep growing. They can actually keep adding workforce, manpower to their company and keep growing. And we have been able to do many of that. Now let's go to my next slide. If you look at this slide, I spend very little time on this slide, but most importantly, what I want to emphasize here is health expenditure per person. If you look at that small number there on the right-hand corner, top corner, you will see India spends about $75 per person. Of this, 70% is private spending, 30% is public spending. So if I want to create a product, I want to create a technology, I want to put a healthcare technology in the market, I have to worry about whether the average, rural Indian is going to be able to afford that technology. If not, then that technology has no value to me. No value to India because, there are many such technologies in the market where, it's only available in tier 1 cities, but rarely does somebody go into tier 2 and tier 3 cities and start thinking of how they can bring a product to the rural market. And and I'm talking both sides. I'm saying cities and I'm saying rural, because I also mean that after tier 3 cities, now you're starting to think of whether this will have an impact in the rural healthcare space. If you create that one criteria in your head and start working on a technology, you have to most importantly work on affordability. And affordability does not come all by itself. It comes because you create a number in your head. With that number, now you start back calculating and seeing how you fit all your components within that number, how you fit everything within that number, and also make sure that the company makes a profit. I'll give a very good example here. I went to London when we got the Wellcome Trust Award, and the Wellcome Trust grant was for affordable healthcare in India. So this was a scheme set up by Wellcome Trust. When I went there, there were all these big people, I don't even remember names now, but many of these large medical companies, had people on the board of Wellcome Trust. And these people spoke from their experience. They said, Rohit, look, if you want to put something in India, put it at 1/3 the cost of the cheapest product in India. So, now let me start giving you examples. So we were working on a urine analyzer, and the urine analyzer, the cheapest urine analyzer in this country is maybe a Chinese made urine analyzer at 50,000 rupees. Siemens sells its urine analyzer, which is called Clinitech, and that sells about 75,000 to a lakh. I cannot put it at 33,000 or 25,000 because that would be taking the higher number into picture. I would like to use the lowest number, which is the Chinese product, and put it at 1/3 the cost so that people would like to use it, not use the Chinese product, and be also happy that there is an indigenous product out there. And that's what gave us a number, and we started working with these numbers. And I'll give you many, many examples. So let me show you some areas that our lab works in. We started off doing magnetic chemohypothermia. We started off because, my first collaboration in 2005 was with Professor Bahadur in MEMS department, and he did a lot of magnetic nanoparticles for hyperthermia. We tied up, started putting these magnetic nanoparticles in gels, in microparticles, in nanoparticles. So, nano and micro multifunctional systems. And we started doing many projects together. We worked on many, many projects over the years. That was one area. But then, we forayed into photothermal therapy for cancer. I'm going to talk about that as well. And that created a big impact for me because that was something very novel. It has terrific potential. And today, there's a company that's interested in putting a couple of $1 million in that technology because they know that that technology has got the potential to reach millions of people in this country. And I'll talk more about that. But then we did a lot of things. We did drug delivery, we did microfluidics. I have an engineering background. I come from VRC Nagpur. Today it's called VNIT, but I still call it VRC. And I have an electronics engineering background. With that electronics engineering background, I went on to do a biomedical engineering master's and a PhD. And I completed both in 4.5 years in the US. And I did not do a postdoc. So I kept myself fresh and came back to India and started my own lab and my own research group in India. I did a lot of things. My MEMS is a core area as education for me. So I picked up microfluidics, I picked up microneedles, and you'll see some examples of those as well. And I also did affordable paper-based diagnostics. Many, many of these that we've done over the years. And we've done many other things, but many of these have potential to move forward. Many of these will lead to patents, will lead to good publications, but they may not lead to actual products. Products are something that one has to work on the separate. So we kept that one area separate in our lab where it was focused on putting products in the market. And that gave us maximum recognition. So all the awards that you saw are because we put products in the market. And there was a lot of emphasis on translation at that time and even today in this government. Now we've forayed into many different areas. So we've gone into protective equipment, COVID care research, because of COVID, there is a lot of emphasis on doing something from each one of our labs. Disinfectant, sterilization products, IoT-based health solutions, because people would like to monitor from a distance. People would like to have everything in their cell phone, and they would still be able to monitor patients in ICU. So we've got several ideas now, many things moving forward, smart dressing materials, critical care products, and many things that are happening in our lab. So we have more than 40-45 people working in our lab, and each one of this student or staff or intern or postdoc has multiple projects to work on. Now, all this started because we put some of these products in the market. Now, when I show this slide, I ask you to focus on all the four sides of the slide, which is all the four lines where you see Dynasense, Mozen, CareNX, Neodox, Norditech, Biosense, Patanjali Pharma, and many others. I have not been able to put all the student companies here. These are all my student companies, and they are startups. I do not hold any stake in any of these companies, so I have no conflict with them except that they're IIT Bombay alumni. But the most important thing is that they each have a product that they have taken somewhere to the market. And when I say products, there are many, many such things. So the journey started. Now let me go to the actual, emphasis of the work, which is showing you, what we've been able to do. So the journey started with UCheck. The one you see in the center is our own urine analyzer. And this urine analyzer created a huge impact in 2010 and 11, because that is when we started off this project. It was finally launched in 2013, but it created such a big impact because it used mobile phone as a diagnostic device in those times when mobile phone had just come out as communication devices. And 2010 really saw a boom in healthcare. 2013, 14, 15, we now started seeing a boom in healthcare products. And this was possible because many of us in different places, different IITs, different institutes, started working on these translational technologies. I'll show you examples. I'll show you details on this as well. But simultaneously, we worked on many other products. The one on the right side is our own glucometer and strips, indigenous glucometer and affordable strips. So each of these, I'll give you a number. I had affordability mapped out so that it was always 1/3, not just the price of the reader, but the price of the strips of the lowest cost available in India. So if today you can buy a glucometer strip, you can buy a glucose sensing strip for 15 rupees or 18 rupees in the Indian market, our strips are costing 5 rupees. And we have been able to do that just because we had this number in mind and we worked backwards. The one on the left-hand side is a smart diagnostic device using conjunctiva of the eye to look at hemoglobin levels. And we've been able to use this device very, very efficiently for several screening camps that we've had now with the student company. And it has actually created a very big impact in this country. There are many others that are following. So we got an Imprint grant to do sodium potassium at point of care, and that became the electrofinder. We also got a Wellcome Trust grant, and that's the left-hand side of your screen, which shows a Smart Sense product. And this was doing the blood, exactly what the ABG analyzers do, but without the PO2 and PCO2. Because that was a little too difficult for us to integrate into this device. So we did potassium, sodium, chloride, bicarbonate, pH, blood, pH, urea, and glucose. And we also had technology for creatinine, so that could easily be integrated in this. The one on the right-hand side was Uritsa, which is 2-parameter, which is using albumin and creatinine. So it's doing an albumin creatinine ratio and looking at kidney markers, basically. Now, how did we start this journey? This is my slide that I show in every meeting I go to, because I emphasize that it does not start. Your solution should not come first. The problem should be defined first. And I always emphasize to all my students, go out, go to a path lab, go to students, go to hospitals, go to clinicians, go to, your general physician and ask him what he faces as problems. If he can give you 10 problems, 5 problems, can you think of now solutions to those problems? And this is where you will have to think very, very carefully, because there will be at least 50 such problems coming out of your survey. Now, can you actually work on all 50 of them? Probably not, which means that now you have to do some sort of a funneling mechanism to figure out which has commercial value, which has, which is the lowest hanging fruit, and which technology you can bring easiest to the market. Now, let me give this example. In this case, we just went and surveyed some labs, and we asked them, what are the tests that they do every day? What are the assays, that they feel they can, get by, that they need from us? What are the instruments that they think that we can make from them? So it was a general questionnaire, and many of these path labs filled that questionnaire. We surveyed more than 50 of these, and when we did, we figured out that many of them kept pointing to urine analyzer, urine dipsticks, and said, look, we cannot afford the Siemens strips, we cannot afford the machine, we cannot afford even the cheapest machine that's out there, and we are not able to do any of this analysis. The problem is, if I'm not able to do all of that, then that means that there is a problem in this country. I am not able to do that because it's not available or it's too expensive. And most of these people said it was too expensive. They're not able to get a throughput. They were getting these Siemens strips in 10 packages of 20, packages of 50. if you open the package, you have to use those strips within a few days. They did not have that much throughput, and they gave us several, numbers and values. So, we started thinking of a solution. I told my student, let's think of a solution. Let's see how we can integrate a very simple solution into this problem. And the problem was there with us. The solution was glaring in front of us because, all this used a, image analysis, and there was nothing to it. All we had to do was use the cell phone, use the camera of the cell phone, use its own image-based CCD array sensor, which is there in all of these mobile phones now, cheapest ones. And we use the cheapest mobile phone. Remember, 5,000 rupees mobile phone, not a 50,000 rupees, not a 1 lakh rupees. A simple 5,000 rupees mobile phone. Use the camera of it. Took images of these strips as they were changing color, put it into an algorithm so that the algorithm would then save these images, the RGB values of these images, at certain time intervals. So if, let me go to this slide. If you see this left-hand side, you see leukocytes, bilirubin, urobilinogen, protein, pH, specific gravity, nitrite, blood, ketones, glucose. These are the 10 parameter urine dipstick. Additionally, you can get 10 plus 2 parameter, which is albumin and creatinine. Now, if you could do the image analysis of these 10 parameter urine dipsticks, because each one of them takes certain time to change color, 30 seconds, 40 seconds, 55 seconds, 1 minute, 1 minute, 15 seconds, so on and so forth. So total over 3 minutes, we were able to take the images of this entire thing, take the RGB values out, compare that to the standard that we had built in the, algorithm curve that we had, and we could actually give you values. When we gave values, we started comparing these values to a commercial system. And in this case, we actually bought a Clinitech instrument in our lab and started comparing it to the Clinitech instrument. And every time we had a student who sat there day in, day out, he was just doing numbers. And we were trying to see where and how you could change your algorithm and make it better. So we had to go through various iteration processes. Started off with a cardboard box, which was an origami design, went on to become a nice box, as you see up in the screen, which became better and better. The first slide that I showed you was the most professional looking design, where the mobile phone is now locked into place, because we saw that people were using the mobile phone and they were not using the box, because they got a mobile phone which they thought was their property, and of course they started using it. But then we had to lock that mobile phone into another version. Finally, we put this in market at 15,000 rupees, because we went through all the calibration, all the optimization, all the validation studies. We did it at KEM hospital in Mumbai, and we made sure that we had enough samples that we tested. We made sure that we had enough data to back it up, and we sent it off for approval to DCGI. In those times, CDSCO still did not have good rules, in place. So DCGI approved this, and we actually got it into market. Finally, when we put it in market, see the prices. The price of the UCheck system is 1/3 the cost of the cheapest system out there. So if the cheapest was 50,000 rupees, which is what our, if you look at that number here, 50,000 rupees, cheapest was 50 because that was a Chinese model. We put it at 15,000 rupees, 1.5. And with that 1.5, we kept telling them, look, if you buy strips from us, of course, you can make it so that it can be universal for any strip. But we said, you buy strips from us and we'll give you these strips at 30 rupees. And nothing was available in market at even 100 rupees in those times, even today. So 30 rupees versus 100 rupees, 15,000 versus 50,000, and 80 rupees ACR strips versus 250 to 500 rupees ACR strips. That created the market for us. That gave us the best, result in the market. The device became commercial. The company started earning revenues, and there was nothing stopping them. This brought us to our second project. And when we started doing the second project, we were called to Delhi by ICMR and asked, now that you have shown, one product in the market, can you now look at indigenous glucometer? And this was in the previous government, not the BJP government, but the Congress government. And they told us, the Minister of Family Welfare, Health and Family Welfare, Ghulam Nabi Azad ji, he had a meeting with us. He told us, look, this is what is needed for this country. And he gave us a number of 2 rupees. Now, this was a very strange request, 18 rupees strips, 30 rupees strips. Even today, you can buy 30 rupees strips because, these people are milking India, Roche, Abbott, Johnson and Johnson, Dexcom, Medtronic, they are milking India because they know India has diabetic population. They know they can charge anything and they will get away with it. But they have the manufacturing capability, so they can actually push these strips in the market. Any new player will struggle to do that. So ICMR called us and said, look, build a team. And they made us in charge of this whole thing. They said, build a team, find people across India who can work on this problem statement, and build an indigenous glucometer and strips, but the strip should not cost more than 2 rupees. Now, this was a dangerous thing for us to accept, but we said, okay, we will try. We don't know whether we will succeed, but we will definitely try. Now, when you're wanting to try that, remember, you have to also look at chemistry. And, when I look at chemistry, it is an open source, so when I talk in terms of software, I'll say it's open source. Because it is off-patent chemistry, there is nothing to it. Glucose, oxygen in the presence of glucose oxidase will give you gluconic acid and hydrogen peroxide. Now, hydrogen peroxide you can use in a secondary reaction with a reduced dye, and it will give you an oxidized dye, which is colored form of that dye. That is easy to detect. It's not difficult. Nobody can patent that. It's there. It's been used in ICMR. All those strips that you saw way back in 1950s, 60s. So there is nothing novel about it. And we said we'll use 2 dyes because you can do a ratiometric analysis. You can take care of interferences. You can take care of variations in the atmosphere, in the instrument, and so on and so forth. So you can actually do a much better reading with 2 of these. So we took up that challenge and started working. And this is what came out as output. So the student was our partner from day one. We did not stop having the student company, because if I had worked from the academic point of view, I would have kept working on the strip, the assay, the optimization for 4 years, and nothing would have come. Now we got a grant from ICMR, and this was a focused grant for 12 months. 12 months ICMR gave us. Can you put a glucometer and a strip, all approved by NIV, NID, National Institute of Design. You had to actually send it to them. And then, you had to make sure that, you got CDSCO or CE approval for that. We went ahead, took up the challenge, started making our strips. You see those strips there, you see the reader. Everything was, industrial design. Everything made sure that, we had beautiful, looking strips, beautiful looking devices, and we sent it out for testing. Now, this is your gold standard testing. It's the Clark's error grade. Clark's error grade will tell you if your readings don't lie in zone A or zone B or zone C. So the one in dark lines right in the center, that's our zone A. 95% of the readings should lie in that zone A. And that's what this graph, this Clark's error grade is showing us. This is the gold standard for glucometers. And you will always compare it to a reference, gold standard in the lab. So you're not comparing it to a portable instrument. No, because portable instruments, like Roche, BD, Johnson & Johnson can all have variations. So you're not going to compare it to that. You're going to compare it to the gold standard. When you do that and find your readings lying between in zone A and more than 95% of that, you're confident your results look good. But we did face a hurdle here. Let me tell you this. We tested it in Maharashtra, right? And in one hospital here, and we sent it to North in Delhi. We had huge variations in results. When it was tested in Delhi in December, huge variations, November and December, and we could not figure that out. Why there was so much variation. But later one of my students actually picked it up that all these problems have different. All these cities have different temperatures, different humidity. Now you have to make sure your chemistry is also stable in those temperatures and humidity. So you have to make sure. We had to go back to the drawing board, do a lot of studies, again send it back, and then make sure that we had 3 centers. We picked one in Chennai as well, and 3 centers where you had data which you could rely upon. Finally, we put it in market. This is what the packaging looked like. You could also do a visual comparison on the strip box. We had these color charts. You could do a visual comparison, but your glucometer was also Bluetooth connected. It was connected to your mobile phone. You could do it. You could put it on the cloud. You could do whatnot with it. So finally, ICMR was very happy. Got released in a very big function in Delhi, and immediately, as soon as it was released, I was approached by 2 large multinationals to sell the technology to them, because they did not want to see this in the country. They did not want to have this technology competing with them. They wanted us to sell them. Of course, we did not, and we went ahead and put it in the market. But It created a big ripple. There was also another technology from BITS, and that also went ahead. But this one, because it had a student company partner right in the beginning, it actually created a bigger impact. And ICMR, went ahead and gave us a lot of accolades for that. Around that time, one of my students submitted a World Cup challenge. So I don't know how many of you are aware, but every year HIT Labs in New York has a healthcare innovation World Cup. And in this World Cup, they invite teams from different countries to submit innovative ideas. And there are many, you can go and look up the website. There are many such ideas, in that World Cup. 160 countries took part in that World Cup in that year. I don't remember I think it was 2013, around that year. 2013, yes. 2013, we went to this World Cup, and we got selected into several rounds, went through multiple screening rounds, and finally ended up in New York. So we had to go to New York, present our idea in front of a large committee, and they liked our idea like anything. Our idea was, can we do point of care diagnostics using a small reader, which can do everything at site? You don't need to send the blood for centrifugation somewhere. You don't need to store it somewhere. You don't need to have chemistry which needs to be added. Everything is in cuvette. Chemistry is in cuvette. Your reader is there. Your centrifuge is on site, and everything is there with you. If you can do that, and if you can make sure that your assay can be done on site in the ICU, imagine the efficiency the doctors will have, the clinicians will have. And we had mentorship from Professor Nikhil Tandon, who is the biggest endocrinologist, in India currently, sitting in AIIMS, New Delhi. He gave us a lot of inputs, and he gave us so much of inputs that, finally we won the third prize in 160 countries. Third prize, 160 countries, and India got a big name in that. But more than name, we then went to Wellcome Trust and said, can you fund this? And Wellcome Trust was extremely happy, with this application, went all the way through. Of course, we got a lot of help from people along the way. So this is why I say, nothing gets done by yourself. You involve people who know their field, who are good in their field. So this is why I say, not just engineering, science is also important. If core science is not there, then engineering has no role, because I will only rely upon a core science to take form. And that's what happened in all these cases. The chemistry was well made, all the device design, everything had a lot of work in it, and Wellcome Trust finally funded it. So that's why I use the logo of Wellcome Trust there, and they funded it. They gave us a number, and they gave us, that look at what is the cheapest analyzer in the market. The cheapest, see, if you look at an ABG analyzer, they go anywhere from 15 to 20 lakh rupees, ABG. Even today, it's not made in India. Even today, it's not made in India. So the cheapest, I don't know what to compare myself. So we took iStat instrument. iStat is a beautiful instrument. It has cartridges, and it is selling for about $4,000 or so. And cartridges are extremely expensive. They're not very cheap. So $4,000, 3 lakh rupees instrument. We said, 1/3 of that, can we build it under 1 lakh rupees? And that was our benchmark. And our benchmark was also, their cartridges are extremely expensive. I think $40, $50. I don't know how much, $50 per cartridge or $40 per cartridge. So we had to keep it really low. We said less than 100 rupees, for our assay. Went ahead and made the reader design, made an industrial design for the reader. Now this is sitting in Wellcome Trust in London. We went and made all these cuvettes. The chemistry is in the cuvettes. We are not adding anything to the cuvette except the serum sample. So you take the blood, spin it down at low speed, take the serum off, and put it into it. So once you do that, you're having a comparison of your, assay that you have in your cuvette, and all this is off patent. Nothing I had to do. You had to just look up the patent for sodium, for potassium, for chloride, for bicarbonate, simple chemistry, and none of that is complicated chemistry. Put it in your cuvette. Make sure that make large batches of it. Put it in cuvettes. Make sure that your cuvette cost and the chemistry cost does not exceed a certain number, so that now you have your consumables to sell and you have a device to sell to the hospitals. And all of this can be done on site. Huge impact, Beautiful data we got from, again, comparing it to KEM hospital. We also sent this to AIMS in New Delhi, and we had good data there. And finally, it was all done, wrapped up, sent to the company. And my misfortune that around the same time that it was sent to the company, which was my student's company, has made so much of money now, has got such big, all these 2-3 products from our lab, and they were making so much money that they were approached by Tulip Diagnostics in Goa for a takeover. And this project got shelved by them. They got taken over two years down the line, because it took a lot of years for them to finalize the deal. 2 years down the line, Tulip bought Biosense Technologies. And finally, Tulip got bought over by PerkinElmer. So today, Biosense is part of PerkinElmer. My students are working for PerkinElmer, and they have the option of actually leaving that and starting their own venture again. But it was a big deal for them. And finally, it created a huge impact in this country. We also did a lot of work. Each one of this is funded by an agency. Many of this, like I said, I work on several ideas. I don't work on one at a time. I work on 10 different ideas, 15 different ideas at a time in the lab. Many of this is not funded. There's internal money that IRCC, IIT Bombay has some money in RDF that I use for doing some POC work, which is proof of concept. If I know that that proof of concept is working and I'm getting good data, now I go to a funding agency and get funding for it. Of course, now most people have seen my face in all these funding agencies, and they don't like to see me coming back with more and more devices. So, it's become more and more difficult for me to get funding. So I'm now going the industry route. So I'm now asking the industry to directly fund these devices, and I'll show you some examples of where we are there. So albumin creatinine, we had shown that with our mobile-based reader. What stopped us from making our hardware reader? And this is our hardware reader. Good design, small strips, all the PCB designing done in our lab, all programming done in our lab, everything, strips, stable, all the assay has been done well. And finally, we were able to compare it to hospital data. Along that same time, we got a Gates Foundation grant, and Gates Foundation gave us money because we went to them and said that, look, this is, of course, this is part of that, but this is the work that Gates Foundation gave, which is why I made my talk drug delivery therapy and all that, because I wanted you to see the flavor of what we do in our lab, which all comes under bioengineering. So whenever I do some work, I start thinking of, I can make a microneedle. Now, let me talk about microneedles here. I can make a microneedle patch. It's simple because it's MEMS. It's in my expertise area. But how do I now combine this microneedle technology with something that makes it smarter? And one of my students and me, we started thinking, and we said, can we make a wearable device? And wearable device has a terrific potential today. You can see all that. Everybody has a smart watch on their wrist. It can do so many things. It can even do ECG monitoring and whatnot. Today, everything is there on your wrist. So we said, can we do drug delivery using these wearables, using these microneedles? And we floated a project, and we got funded by Gates Foundation, and we started working on it. So this was a silicon microneedle based project. The first one I showed you was a polymer based. So there's a lot of interest. This became an offshoot of that project. And we now have capability of making polymer needles, microneedles, any height you tell us, and you look at the microneedles that are shown in the SEM image that you see, beautiful microneedles, pointed tips, which can be loaded with drugs, with biologics, with whatever you want, and it can release that in the body and get dissolved completely. Imagine the potential of these microneedles. Imagine how you can use these microneedles for vaccine delivery, for I don't know what not, what applications. So people coming to us with other applications. This is where we started, and it was actually a difficult project for us, because initially when I started this in IIT Bombay in those times, in 2010, 12, 13, 14, I think 14, 10 or 11, there was no DRI facility in IIT Bombay, and which is deep reactive ion etching, which is used to create these tall structures. There was only a reactive ion etching process. With reactive ion etching process, you cannot get tall structures. You can get shorter, height structures if you're using silicon micromachining processes. With this, we were able to show that we could get tall structures, and we actually patented the process. Went on to show microneedles, which had similar dimensions as our 34 gauge needles. And, they were as thin, as sharp as these. Of course, you don't see the sharpness here, but since they are small and thin, you can just press them into the skin. I'll give an anecdote here. I know this was also funded by NP Mass for just the microneedles, NP Mass, in Bangalore. And we went there, I had to present these microneedle patches to the committee. And there was this big committee sitting there. And I was asked to present. I was presenting this data. Everybody asked me, can you show me microneedles? So I had taken some patches along with me. Now, these people, all these committee members, imagine what they're doing. They're taking these microneedles and pushing them into their skin just to see whether it has strength. And it went in. Of course, whether it broke inside, whether it did not, whether it stayed inside, I don't know anything about that. But the fact is, I did not ask them to put it, but they did. And it actually withstood the pressure with which one pushes. And it actually stayed, and it proved that our microneedles had strength. Now, how do you make a smart device? Simple to do. You take a watch apart, see what is there in that, and you can easily make it. It's not a big deal. Reverse engineering is something that I can, most of us can do. And it does not take rocket science to do all that. You can make this smart device. But how do you combine the microneedle at the base is where some engineering principles came into play. We were able to do that. This is the final design that you see with the microneedle at the base of our watch-like device. But to get here, we had to go through several iterations. So I show this slide and I show you all the iterations. And the iterations are that you have to actually go work with a larger prototype. Because, see, your industrial design comes later. But your initial, version 1, version 2, version 3, they come, first. So these versions had larger pumping mechanisms. These versions had larger form factor and larger reservoirs. But we were able to show drug delivery profiles which look like what you see at the bottom of your screen, right-hand corner of your screen. And this was terrific data that we showed to everybody. And people were very happy with this data. Initially, we showed ondansetron release, and we said, can we do it for people who are undergoing chemotherapy so that they don't have to take these drugs through the oral route? They can actually take it through this route. And this is a beautiful idea in that sense. We patented that idea, went ahead, and there were many companies interested in it. Today, again, there are many companies which are interested in this technology. And we are hoping that there will be some of this moving forward. Around that time, I have been sitting in BIRAC panels for last 12 years. And all ICMR, DBT, DST panels, I'm part of every panel in this country. Everywhere where there is translational research happening, where there is bioengineering. And it's not because I'm good, bad, that the committee, rarely sees, if you are the best. No, they see whether you have expertise in that area, whether you have experience in that area that you can help others. And that's what I have been doing. So sitting in BIRAC, I know the ecosystem, I know what companies are doing what, and where are the gaps. And this came to us from Professor Padmanabhan, who chairs the BIRAC committee. And he told me, Rohit, everybody does. And I was at this NASI meeting where I was getting the NASI Reliance Industries Platinum Jubilee Award, and I was presenting my data. And he said, Rohit, everybody does what you show. Urine, glucose, everybody does. I have not seen a lipid profile anywhere. I have not seen some of these important cardiac markers anywhere. I have not seen, liver profile anywhere. Can you think of some of these? So I said, sir, that's a very good point. I don't know whether India has such products. At that point, even I was unaware that there were anything in the market. Went ahead, did a good literature search, put a student on it, found there is nothing. There was this call check instrument which is available in US. It is available on Amazon. You can actually see on Amazon cardio check. Call check is our own, sorry. So call check is our own device, but cardio check is available in Amazon. And this is extremely expensive device, not sold in India. You can buy it on Amazon.com. Whether they sell it to you, whether it comes to India, I don't know. But the strips are expensive and it's extremely difficult to do. So all we had to do was look at what they were doing. They were doing total cholesterol, HDL, LDL. One of this is using a formula, tritol and triglycerides, TGL. Now, you look at the chemistry. This is the chemistry given here. Nothing rocket science. Chemistry is out there. This uses 3 enzymes. The other one, the TGL uses 4 enzymes. 3, 4 enzymes. How difficult it would be. So, of course, we said we'll pick up the challenge. We went to BIRAC, floated a project to BIRAC with enough preliminary work done. Because see, when I go to BIRAC, I always have proof of concept done. I never go with an idea. And that is something that I do tell everybody, each one of my colleagues, please have some proof of concept done so that BIRAC also understands that you have the capability of doing that. You've done projects in that area and you can now take this project to completion. So sitting in that panel, of course, we could understand what came to those panels, but still I had to present as an outsider. I did present that, made sure that the committee liked the idea and said that, look, we'll make two versions of the device. One version would sit with the doctor, with the path lab, small version in tier 2, tier 3 cities. One version you can carry home, or even the doctor can have in his bag, which is the paper-based version, which has more stability and has more shelf life. The liquid assay-based version, you have to actually keep it sitting somewhere, will have less shelf life because you're using liquid chemistry, but then you'll have to need the use of a fridge and all that. So, of course, you can't use it anywhere or carry it in your bag. Two versions, and we said we'll make a device out of it. So we started working on chemistry. We had done the liquid chemistry. Liquid chemistry is simple. You mix all these enzymes together and in whatever ratios they are needed, have the dye with it, and it works. And the difficult part was making the reader. So we had a student company from the beginning, Diagnostic Technologies, which worked on the industrial design, worked on the design for both the liquid assay reader, which is the bottom side, and the paper-based assay reader, which is the top one. Both of these designs went through several iterations. Finally, we fit them. Finally, we fixed them and moved ahead and started doing testing with our devices, with our assay, with our stability, with the readers that we have, and comparing it to, again, hospital data. So every time there's clinical validation being done at hospital samples, we are comparing it what is sitting in the hospital with our device. And we had less deviation, less than 10%, 5%, and 10%, in some cases a little bit more, but then that was just because of the range of the value that was chosen. But still it is fine because, for a point of care reader, anything above 85% is good. So, if you're getting 90% and above, you will get approval. I don't know how many of you know, but COVID antibody tests today, which are there in market, how much the sensitivity of these antibody tests are, or antigen tests are, 50%. ICMR has put a limit of 50%. If you get anything above 50%, ICMR will approve that test. Imagine that 50%, one of two is going to be wrong, which is why you see so many erroneous readings. And I sit in all of these committees, and I am extremely sad that people come with 55% and get an approval. Whereas, if somebody comes with an 85 and 90%, I'll be very happy with that data. So this is exactly what that data was. We went back to BIRAC, got a CRS grant, which is coming back with a company partner. Of course, we had a company partner in the beginning, but the company partner was not taking money out of it. In the second phase of the grant, now the company partner gets money. And this money he can use for commercialization, for scale up, for outsourcing or whatever, and he can make that. He has to have an ISO facility, for manufacturing. And then he has to make sure that manufacturing happens. Today, we have made these readers. We have made our assay chemistry in these boxes. We have our strips, and each of this has been contract manufactured through the company partner. It uses less than 40 microliter of blood sample directly onto your strip. And, it has a filter. It will actually filter that out. And it will give you readings of total cholesterol, HDL, LDL, and triglycerides. Simple device. It has tremendous potential. The liquid-based assay, a reader also, we are moving forward now through CRS. Similarly, we got a product into ICMR, funded by ICMR, for doing troponin I and troponin T. This is using lateral flow assays. Many of you are aware lateral flow assays are there in market. Many of these lateral flow assays are there, but very difficult to read them. We figured out that you could do reading of these lateral flow assays if you do look at intensity of the test line and control. The control line is always lit up, right? And that shows that your test is working. The test line is lit up only when the analyte is present. Now, if you have a specific antibody to the antigen that is coming in, or your analyte that is coming in, it'll light up. But the concentration also makes a difference, because the deeper it gets, the more concentrated sample comes in, or the concentration keeps increasing. So you can do a nice analysis. We made a small mobile reader. We used the pregnancy strips to do all our calibration and different concentrations of the hormone. But the fact is that you can actually use it for any other. So we've started working on A1C, we started working on vitamin D, we started working on T3, T4, TSH, so thyroid markers, started working on many things using lateral flow assays. And we are getting good results in many of these because making the strips is not difficult. Reading the strips is the difficult part. So this is where we are in many of these, and I hope 2 years down the line, I'll have 1.5 years down the line, I'll have enough of the data to show all of you that many of this is in the market. So this is what our A1C chemistry looks like. Chemistry, of course, I am not going to disclose the chemistry here because it is with the industry partner. But finally, they have been able to make what we could do in our lab, translate the chemistry into a small reader and a strip. And this will use very low amount of blood, but will give you your A1C value, which is looking at the 3-month, diabetes marker. So I can do 3-month. I have also a marker for 3 weeks, which is glycated albumin, and I also have spot glucose. So we are now putting it all together into a glycemic panel, which will tell you all and of course, everybody will have a different market. The urine analyzer, we went ahead and made a different version for it. Sorry. And now it can do 14 parameters. So we've also added a couple more parameters, calcium, as you see, and microalbumin was there, but calcium and I think ascorbic acid has been added into this one. And it actually is giving us terrific results because now we are comparing. There's a student who set up a company around this technology, and they've already become a 100 crore company. So one of my students set up a company about 6 months back. They have raised enough money. They're already valued at 100 crores now. We are also into many other things. So a student has come in from Indian Navy, medical doctor in my lab who's doing a PhD in my lab. He and I have been working on several technologies now, of course, for using silver nanoparticles, and we are now getting terrific data. So we have now technology to move this into company. Many things happening in our lab. I cannot even give you snapshots of what we do. I'm going to go into the photothermal side as well. But we do all this in our lab. So we've got our own bone screws, which are, of course, biodegradable, less than 1,000 rupees, and we've set up a company around that technology as well. Me, Professor Belare, and 2 of our students. We've got a technology for doing periprosthetic infections, which is CRP and IL-6. And CRP now has a lot of interest. So we're looking at CRP as an inflammatory marker, ferritin as an inflammatory marker, and D-dimer as an inflammatory marker. In COVID, this has become a beautiful marker set. If you can have a point of care device which can do all 3 of this at bedside, imagine how many patient lives you can save because you can immediately tell them this is how your inflammation is increasing. You now need to take preventive measures. We are also focusing on creating affordable healthcare labs using nanotechnology as the enabler. So my lab started off with nanotechnology. I'm not going to leave it. And there are many things that are happening. So we've got several patents filed in nanotechnology domain. And each one of this now has tremendous potential to be translated. So we've set up a company. We're setting up a company around our photothermal therapy in cancer. We're setting up a company around already set up the nano-engineered biodegradable bone and tissue fixation devices. We are setting up a company around our drug-loaded nano-engineered sponges and on-demand drug delivery system. So there is a lot of emphasis on many of these things which use nanotechnology. This is a snapshot of what we do with nanotechnology. This has huge potential. This got published in Nano Letters. I don't know how much time I have, so about 5 o'clock. So I'll finish in 5-7 minutes now. So we published this data in Nano Letters, and this created such a big impact because we showed that using these gold-coated liposome nanoparticles, you could shine near-infrared light on it. This near-infrared light, when it hits these gold nanoshells, would convert the incoming light energy into heat energy using surface plasmon resonance, and they would then create enough local heat to kill the cancer cells on the site. And this was shown not just in vitro model, but several in vivo models. And when we went on to show in vivo clearance model of these nanoparticles, because when you're hitting them with laser light at 808 nanometers, the core is also heating up. The core is a biodegradable, thermoresponsive lipid core. And this heats up. When it heats up, it breaks. As it breaks, the shell which is coating the core also breaks into sub 5 nanometer particles. these sub 5 nanometer particles now get released and they get excreted by the body through the renal route, and they are not there in the body. So we could pick them up in urine and in feces. And imagine if this is possible. Imagine the advantage. There is this company that is doing it. I just go back one slide. Orolase therapy. This is Naomi Hallis's group in Rice University that's doing this work. They have a silica core with gold on top, and they have put it in phase 2 trials, and it has failed because the silica is non-biodegradable. They could not solve that problem. We solved that problem. We patented that technology, and we made sure that it moved ahead. Now today, with GMP manufacturing happening in our department through a alumni network, alumni is giving money to set up GMP facility. We will scale up the material and start clinical trials at Actrec Mumbai. I do all of this with Abhijit De's group at Actrec, and I hope that we can do it at Tata Memorial, because we now have tie-ups with several people who are interested in taking this technology forward. Most importantly, look at the data. 1 dose, 2 doses, maximum 2 doses, and the tumor is gone. Doesn't come back even after 180 days. And there are a lot of data we have in this, and it has good toxicity profile, acute, chronic, both of them we did. And we showed several things with different models. So now we are hoping we'll take it forward. We also do a lot of work in theranostics. I can't even go into all that, but many work happening in theranostics where we do, imaging as well as therapy. We've developed our own bone screws, as you can see, and these bone screws, and I'll give you just one example here. The one in the market, which is biodegradable bone screws, the one which sells in the market is Smith & Nephew screws. These cost 15,000 rupees. So we bought these, saw how they were made, and they use PLLA HA. So they use patented material. You can't use those materials because they will actually file big suits on you. So we had to use a different material. And there are these big companies. See those companies: Stryker, Covidien, ConMed, and all these companies, Smith & Nephew. They'll be after you if you try to get into their space. So we said we'll not get into PLLA. We'll start working with different materials. So one of my PhD students took up this project with Professor Bellare as co-guide, and we did silk magnesium oxide PCL. And we did not use PLLA, we use PCL, which is polycaprolactone, which has slower release profile, but you can control the release profile and you can actually make sure that it degrades. But then you have to give it enough strength so that you can replace the screw that they're using in the market. So our screws turned out to be better. When you go and do all the testing, we did all the strength testing. I got loads and loads of data. All of this is one thesis. And each one of these projects that I'm showing you are 100 slide presentations. I'm just giving you snapshots. So each one of this work, actually led to a company. Because there was potential, we had filed patents, we made sure that there was novelty, and we made sure that there was applicability of this. Today, if I can put these bone screws in the market, imagine the impact it will create. Somebody buying them at 15,000 rupees, can they get it at 3,000 rupees? 1/5 the cost, or 1/6 the cost. Can they get it? Because I can make it at 100 rupees, so I can sell it at 1,000 rupees. Now, of course, the company will need to make some profit out of it, so we'll have to work the prices out. I don't know what prices. Finally, we put it in the market at. But they have a lot of advantages. Then we also did orthopedic surgical site and implant associated infection prevention. And this came from a clinician, a surgeon who visits our lab, Dr. Arun Mullaji, one of the biggest knee replacement surgeon in this country. He has shown us a lot of work. He showed how the data, he showed what exists in the market and how you can make a difference. We went on to make our own patches, and these are made out of chitosan, used them, drug loading in them, microparticles with drug loading, showed antibacterial studies, drugs, given by the clinician, vancomycin, cefuroxime, ciprofloxacin, 3 different drugs, 3 different case scenarios, all different sponges, degradable sponges. At the end of it, the sponge is gone. 3 weeks, sponge is gone. Different materials and beautiful data. Again, this is being picked up by a company which is now saying that they would like to put some more additional functionality to this and put it into the market. With that, we also started looking at social innovation from the lab. We said we've done a lot of commercial work, done a lot of work for students. Can we do something for the country? And I said, I'll pick up something which is a big challenge for this country: anemia and malnutrition. If I can target these two biggest problems for this country, and I have a solution for that, or I have some sort of data generated for the government, maybe that will create a big difference in mindset of people. So we started working with that, took up my student company's products. The bottom one that you see is Care Mother, which is from a student's company, which is doing maternal screening. This is 0 to 9 month screening of pregnant women in slums, in rural areas, in northeast corners of this country, and where not. But he has been able to make an impact because if he finds high risk pregnancies, these are referred to a primary care physician. That person does an intervention and one life is saved. So we have been able to save more than, 10,000 lives till date. They have screened more than 1 lakh women now, maybe more than that. I don't know the current numbers. We also started doing malnutrition screening because, we made a smart using technology. You can do height and weight measurements using smart technologies. You can do anemia screening using the touch. We started taking all these products forward into many, many camps. This is what the Care Mother has, all these different parameters and everything on your mobile phone, all app connected, everything in a data, and everything uploads on a cloud server. Did that in several places, in Anganwadis, in slums, in several places. In Churu district in Rajasthan, the students company went and did a lot of screening, and we found good data which we could give to the government, which we could show to the government that, look, this is the SAM-MAM status in this district. Can you do an intervention so that these numbers come down? Can we do an intervention? Not an intervention, we can do that. Monitoring over a period of time. Can we do it now? Can we do it 6 months later, 1 year later, 2 years later, and show you data? And can you do something? And this is in Rajasthan, anemic women, many lives saved because we gave data. This is for Care Mother. Again, several villages reached, many mothers served. Again, this is old data. I have to ask Shantanu for new data. Shantanu is doing a PhD in my lab, and he's already working very hard with this company. He's got commercial success, has presented at several places. With that, let me come to the last 3 slides of my presentation. Let me thank all the funding agencies. Nothing possible unless you get the best funding possible. Bill and Melinda Gates, Wellcome Trust, Grand Challenges Canada, we got twice, and this is very, very difficult grant. BIRAC funding, very difficult, very competitive. Imprint funding, we got 3 of those. Many difficult grants, but most importantly, with all this, we were able to show output. We had students who were graduating continuously. I never stopped taking students. Even today, my lab has 22 PhD students. Can you imagine the number? I have 22 PhD students. I have got 8 master students. I've got 10 postdocs, and I don't know how many interns. 45, 50 people today in my lab. And we keep training interns because I want them to see what this country has to offer to you. If their college does not have the facility, IIT Bombay has it, and we will make sure that you get trained on it. We will make sure that we give you a letter of recommendation so that you will do well in your career. And we do not say no unless the IIT system says no to us. And we have trained these people. We have also given help to many innovators. Most importantly, said right in the beginning, we do not stop filing patents. More than 120 filed from my lab alone. There are many colleges in this country that have not filed more than 20. So, if one lab can do all this, why not 400 other labs do the same model? And that's what I keep telling everybody. Anywhere I go, I say, look, if I can do that, why don't you do that? Why keep your numbers limited to what IIT tells you to do? IIT says 8 PhD students max. I've always had 24 PhD students in my lab. I have never worried about numbers. I've never worried about rules. I've never worried about what somebody tells me to do. I only worry about output. If my output shows up there, I'm sure somebody will get benefit. Products have been commercialized. Startups have been created. High value jobs have been created. Patients have been benefited. That is my biggest, satisfaction in this country, that we have been able to benefit somebody. We have showcased this work to several people in this country. Of course, the Honorable Prime Minister was there at IIT Bombay. Loved what we were doing. The HRD Minister, the Science and Technology Minister, everybody has seen what we do. And there is no dearth of showcasing our work. We showcase our work to students, to Kendriya Vidyalaya students, to college students, to everybody, who comes, because we want them to see what is the possibility of bioengineering and healthcare. And we have tie-ups with all major hospitals, with IITs, with student companies, with large companies, with incubators. I am on most incubators panel. I chair the BIRAC big panel for diagnostics. And I have a lot of good connects now so that I can push these people into places where they can actually make their product. Let me end this by thanking these three people who've made a difference in my life. Dr. Mrs. Manju Sharma, who has been a mentor to all of us, all the youngsters in this country, but more specifically to me, because, she has helped me at every point in my career and shown me, what this country needs and where I need to go. Professor Padmanabhan, who chairs BIRAC, nobody not knows him. Everybody knows him. And he has been a pillar of strength to all of us. And Dr. Rohit, who has given us the insights of what this country needs and how BIRAC and can help us. He has also been a strong support. All these people. Thanks to all the faculty members. Thank you very much.
Srivastava, we can't hear you properly.
Yeah, so I am saying thank you to everybody, but thank you to all my mentors, mainly Dr. Manju Sharma, Professor Padmanabhan, and Dr. Suru. I think there is nobody bigger than these people who I can thank, and they have made a difference in my life. They have made a difference in thousands of lives in this country, millions of lives, and I think we can keep learning from our mentors. Thank you very much.
Thank you, Professor Rohit. So it was a fantastic lecture, and as you said, I think every part, every product, every idea, every initiative, I think merits a colloquium on its own. But of course, you gave such a big canvas on which you and your department and several companies that you spawned has actually worked and continue to work, and the results are all there, ultimately benefiting the humanity at large and creating a lot of value by way of entrepreneurs, and also producing fantastic value to this country as a whole in this field. And I mean, the reach was fantastic. I mean, it's not just some very specific area, and you really covered a large canvas. So thank you very much. And I'm sure there are quite a few questions waiting for you. Let me, so there is a hand raised. Yeah, Mr. Vivek Datar.
I wanted to ask you about a diagnostic for Chikungunya. This is a reasonably common, ailment, and it affects people for a very long time. I mean, sometimes up to 6 months and so on, and pain at various. So, is there a good, because right now the diagnostic, as far as I understand, is something like 4,000 to 5,000 rupees in some of the standard labs. Can that be brought down, to maybe even a 10th of that level or a 5th of that level?
100%, sir. With all these different technologies that have actually gained ground now with COVID, and people have been able to bring down the RT-PCR cost from 4,500 to 450 rupees today. And if that is possible, I'm pretty sure all these others will follow suit. Today, people are working only on COVID, but very soon you will start seeing, with one technology now in place, they will now start using the same ones for all the other, diseases that you see, and you'll start seeing prices come down. I'm pretty sure it is just around the corner.
Okay, Arnab, I thought you have a question. Do you want to ask? Or do you want me to read for you?
I can ask.
Yeah, please go ahead.
First thing, thank you. This was such an inspiring talk. I think that's the word I'd like to use.
Thank you very much.
What you have achieved. I just want to put another question. I mean, often you are saying, okay, 1/3 price point is where we should do it, and that's a good place to go. Do you sometimes face, you did say that, the big players want to buy you out just so that they can not use your technology. But do you sometimes have a problem which we often face, that people think, "Arey yaar, itna sasta hai, so it must not be good. The more expensive one is good."
So I'll give an answer to that, Professor. The answer is, if you focus your products to tier 1 cities, this is what you will see. This exactly what you said, because people say that it is like a Chinese product and it has no value. Okay? But the moment you go to tier 2 and tier 3 cities, there is so much of value to pricing that the moment you bring it to that pricing level, now everybody starts thinking that, look, this is a product I can actually buy and put in my path lab, or I can put it, in the clinic. So I am saying this right from beginning. I don't want to target any of the tier 1 cities because I know it's not going to sell there. And all my product is going to be targeted tier 2, tier 3 and rural. And rural, the best part is that unless you create anything less than 10 rupees, it won't sell. So, whatever product you make, if it is not less than 10 rupees, there will be no buyer for it. So I have to think in that sense as well.
Great. I might approach you later for some ideas which we had with Tata Memorial, but I'll come back to you later with that.
Sure.
Thank you very much.
Thank you very much, sir.
Yeah, thank you Arnav for that question. I thought actually similar question was also posed by Prof. Vivek Datar here on the chat window. Are there products which are even cheaper than 1/3, say one?
Of course.
150.
Of course. So drugs are, I'm not comparing drugs because see, India is the major player in now all this drug business. So, pharma companies, that is why they have made so much money, because anything that went off patent, now they are making in India and bringing it extremely cheap to Indian people. What I am saying is all these diagnostic products, and you were right in the beginning as well, for Chikungunya, if people are pricing it at 2,000, 3,000, 4,000 rupees, not many people are willing to buy that. If we can bring it to under, say, 200 rupees, then you will start seeing, a lot of people buying it. So that's my goal. I don't want to create it so cheap that, it looks like, like some, the previous person said, Chinese product. But if I target tier 1 cities, I have to keep a price range where it will sell. So Wellcome Trust also had many inputs around that. They said, if you target tier 1 city, do not go less than half of what the best product in the market is. Not the Chinese product, but the best product in the market. Do not go less than half of that value in tier 1 city. Tier 2, tier 3, this is the other formula. That's what I had to say.
Yeah, I guess Professor Datar has one more question. He raised his hand. So.
Yeah, this question, I was quite excited by your, the boards that you have made, which are biodegradable for bones and so on.
Yes.
So I just wanted to ask, is there any possibility of, these tooth implants, which are right now, I think, made of some, very expensive material, titanium and stuff like that, which I think each tooth probably costs about 1 lakh or something.
Yeah.
Is there any possibility of making that much cheaper? Because, there are many people who unfortunately have bad teeth and, they drop off and then they have to have some solution.
So actually, sir, what we are doing is there is this different area. So instead of making implants, Professor Belare's group and me, we have set up this company where we are using tissue engineering to make implants. So we have made a scaffold, which, based on the cavity size, that scaffold will be implanted, and that scaffold will promote cell growth. It will actually be filled with, your cell growth factors. It will actually promote cell growth and make sure that, we don't need to put an implant in place. The bone itself regrows to that point.
I see.
So there is this way that is happening, sir.
I see. Very interesting. Anyway, I didn't mention it, but I think I was quite excited by your talk and the range and the reach of your
Thank you very much.
Research and the products that you have. Thank you very much.
Yeah, I think that's the kind of feeling we all are sharing. It's a great talk with lots and lots of throughput. I mean, the statistics that you have been saying about your group and your students and your products and so on are boggling. So if there are no more questions, and let us once again thank Rohit Srivastava, and we wish him many, many more success stories. More importantly, the service that he and his group have been doing to the community is unparalleled. So thank you very much, Rohit, and thanks a lot. And looking forward to many other success stories in the future.
Thank you very much, sir. Thank you so much. Thank you.